Even though the discovery of nuclein by Friedrich Mischer and the proposition of principles of inheritance by Mendel were almost at the same time, but that the DNA acted as a genetic material took long to be discovered and proven. Based on Mendel's observations on Pisum sativum, he proposed that something was being stably passed down, unchanged from parent to progeny over successive generations. He called these things as factors. Now we call them as genes. In early 20th century, it was hypothesized by a number of scientists that proteins are the substance of heredity, while DNA had been known to science since 1869. Also, it was believed that DNA has a restricted role within the nucleus, but an experiment by Avery, McLeod, and McCarthy in 1944 first suggested that proteins are not the substance of heredity as previously thought, and instead DNA was the culprit. And the situation was like everyone expecting the brilliant student protein will top the class, but in reality, a backbencher DNA, not even considered in competition, becomes topper of the class that is acted as genetic material. However, given that every young colleague's results flew in the face of current thinking, not everyone was convinced. In science, a great deal of evidence is needed before a new scientific idea is accepted as fact. Therefore, in 1952, Alfred Hershey and Martha Chase carried out experiments in famous Cold Spring Harbor Laboratory, USA, which addressed the same question: was DNA the hereditary material, or was it protein, or was it something else? To solve this important puzzle, Hershey and Chase needed a simple model organism to solve this complex question and a clever experiment. They chose the simplest organism possible, a bacteriophage, which is a virus that infects bacteria. This virus looks a bit like a lunar landing module and attaches outside of the bacteria. It then injects a substance into the bacteria to create many new copies of virus. These newly manufactured viruses are released when the bacteria bursts. So the substance injected inside the bacteria must contain the instruction manual for making new viruses. It contains the hereditary material. But what was this material that the virus injected into host? The answer to their question might shed light on the nature of heredity for all life on Earth. Viruses consist of two things: a DNA or RNA core surrounded by a protein coat. Hershey and Chase needed to figure out which of the two was injected into bacteria. They ran two experiments, and in each they labeled one of the components. For this labeling, they used radioactive tags: radioactive phosphate for DNA and radioactive sulfur for proteins. This was an elegant method because phosphate is only found in DNA and not in amino acids that make up proteins, and sulfur is only found in proteins and not in DNA. After the viruses infected the bacteria, the researchers examined whether the radioactive signal was detected inside the bacteria or was left outside. To do so, they needed to separate the bacteria from viruses attached to their outside, and did so using a varying blender. Once separated, they then centrifuged the sample. Since the bacteria were bigger, they were centrifuged to the bottom, while the smaller viruses remained at the top of the tube. When they prepared viruses with sulfur-labeled protein and mixed it with bacteria, they found that most of the radioactivity remained within the virus and did not enter the bacteria. When they allowed the virus to replicate and collected the new viruses, less than 1% of the protein radioactivity from the viral parents was found in the newly replicated virus. So the protein was not being inherited. On the other hand, when they performed the same experiment using virus with its DNA labeled with phosphate, they found that most of the radioactive DNA was injected into bacteria from the attacking virus, and much of this labeled DNA was inherited and passed on to the new virus generation. Hershey and Chase added to the mounting evidence, pointing the finger at DNA as hereditary material. From their observation, they stated that DNA had some function in multiplication of the bacteria, whereas proteins did not. Hershey and Chase experiments spread quickly and helped to convince many scientists that DNA indeed was the molecule of heredity, and the remaining skeptics could not hold out for much longer. A year later, Watson and Crick published their DNA double helix model, which provided an elegant explanation for how DNA with such a simple structure can be replicated and pass along information from parents to progeny. The Hershey and Chase experiment is considered a classic in biology because it tackled an important question: with the use of well-chosen model organism and elegant experimental design to clearly distinguish between the two competing hypotheses.